All right. I hope you guys are excited for this afternoon. Are you ready to hear some stories? Thank you. Thank you. Thank you. Yes. I know we're very excited to hear some stories and create more community here. This is a mission for the GGA is getting more personal stories, or we like to say sometimes science adjacent personal stories. It's been told to me before in one of the trainings I did on storytelling that we like to think of each individual as a swatch of fabric. And actually, the stories we tell is the thread that links those fabric together to build something beautiful, like a beautiful quilt. And so we get a real treat today. We have 5 amazing storytellers. So the way this is going to work is I'm going to briefly introduce the storytellers. They're going to come up and tell their story, and we're going to give them a lot of support, and then they're going to sit down and the next storyteller will come up. We don't really do question and answer periods with stories. Y'all ready? All right. Our very first storyteller is Carlos Goller. Carlos is a microbiologist and a biology educator in the Department of Biological Sciences and teaches in the biotechnology program. He grew up in San Miguel de Allende, Mexico, and uses molecular biotechnology and sequencing technologies to learn about microbial processes and sustainability. With molecular techniques, Carlos and his students learn about the microbial communities in compost, kitchen sinks, waxworm guts, and bear scat. And he works on bringing high throughput technologies and robots to courses. Carlos also enjoys working with faculty on biology, education, research projects that span multiple institutions. When he's not sequencing microbes or bear scat, teaching courses, or experimenting with new instruments, Carlos enjoys time with his 2 kids, amazing partner and educator, Claire Gordy, who we heard from earlier today, and his dog, Sunshine. Carlos.
Okay. Thank you for this opportunity and challenge. What did I get myself into? At least it's after lunch, so we will begin. And I'm setting a timer so that I remember. So, after lunch, picture this. Hanging from thin threads, slices of bread suspended in the air by paper clips. Different slices have different gooey, mossy, rich mold. This is not the biology lab or microbiology lab. This is my high school apartment. Now picture my mother's face when she dropped by to surprise me with food, entered the room, and saw my assortment of different breads and mold hanging in there that I missed. I walked by with a mister and missed every day for a week or 2. So my mom was shocked. Yet this was one of the most meaningful experiments I've done. This was an experiment I did as part of a high school class with a fantastic educator. Gabriela Corral was a teacher, is a teacher, and taught this class in English in Mexico in a high school called experimental design. And this was one of the class challenges to design an experiment and complete it. I just brought it home. And in this class, we were challenged, yet given the flexibility to learn about the scientific method. And I really loved that. And in contrast, the room across the hall the same class was taught in Spanish by a renowned professor, and it was taught by reading papers and thinking about the steps. Whereas Gabby made us think about HIV and Ebola and this outbreak that was happening and this cool thing called biotechnology. And that last word, biotechnology, really made me think. I'm like, wow, this is really neat. What can I do with slices of bread and that mold? And Gabby inspired us. Yet, even before this, I had excellent mentors. I grew up in a small colonial town, now a city. My dad was retired from the Navy and taught others to fly planes and land on aircraft. My mom is a school teacher. And it was a city full of contrasts. So was I learned board games with a German couple across the street. I played with cars in the US consulate's garden. I may have put a ladder to go into the neighbor's pool. The neighbor's pool was awesome because they were on the poor side of the Vanderbilt family. So I had a fun experience growing up and learning from different yet contrasting people. I learned about experiments because someone who gave me a microscope was this German couple, and he was Konrad Plumpe, was a prisoner of war. Yet he taught me how to play with a microscope. My mother always encouraged me to experiment, and my dad was always learning. Even after being flown from Mexico to a US naval base to seek medical attention, my dad was teaching the pilot how to communicate to land in a military establishment with a private Cessna and was saluted upon arrival. So I had really great role models. Yet I in high school I had a hard time because in high school I realized that I was lucky to be in a private high school where I had a fantastic set of professors that allowed me to be creative. Yet it was a world where outside of the classroom we had two bodyguards protecting the daughter of the CEO of Coke. And I was a work study student that in order to keep my scholarship, I had to be a grader. And I would wipe these round plastic things that were part of computer mice in the computer room and learn about Google searches or Netscape searches back then. And I was searching for biotechnology and Ebola. And this prompted me to say, I want to go and study biotechnology. And I applied only to colleges that had biotechnology, ended up at Worcester Polytechnic Institute, and loved it. And I did that because I took some risks. I said, no, biotechnology is a major. I can do this. Much to my mother's dismay, I printed everything out on recycled paper because I was going to make a point. And I said, well, they could read my drafts if they want. They get my personal statement and my homework from class. This is awesome. And in college, I had roommates and mentors who were really contrasting. For example, one mentor is a traditional biochemist, has been on NIH panels for a long time, really taught me how to do things. And the other mentor said, you know, we should go out to Cape Cod and get some fish and figure out this RNA thing. And then maybe we can do some tagging and look at the expression levels. And the same at Emory. I had an encyclopedia of E. coli. That was my mentor. And a wacky professor who brought in the Dalai Lama for a series of workshops. And they both mentored me. As a postdoc, I had a fantastic MD, PhD mentor, and also a creative educator. And now that I recollect and think back about this, I see, what did I learn from these? I learned that I really value the mentorship and that mentors are diverse and that there is in my world, in how I work with students now, I appreciate the flexibility. But I also think, what are the costs of this? Am I doing a disservice to those I mentor by being a little more flexible? When I hear the words rigor and professional practice, they make me shiver a little bit inside. And I find myself being very different as a mentor now, and reflecting on that contrast between Gabriela Corral and that professor. Gabriela Corral taught dozens of students in English. We've got a couple of Stanford professors, we've got physicists, we've got musicians, we've got one or two actors from that bunch. And the office of that other professor is stacked full of manuscripts, but is no longer open to students. And he's no longer teaching that class. And I wonder, how many times did they get the opportunity to do something like this? Or get the excitement of doing some Google searches and learning about Ebola? And that flexibility to be able to take their curiosity and find a path and say, I'm going to study biotechnology. And we think back about this as a cohort because many years later, we still keep in touch. And Gabriela Corral ended up going up in administration and becoming a chancellor for education and education reform. And her message was really powerful. And that experiment was really fun. Even though it shocked my mom, it started a process in me of really thinking about my favorite phrase as a 5-year-old. Don't be so sure. My dad suffered a stroke. I said, Mom, is he going to be okay? We didn't know. And my mom said, maybe he'll be able to uh, to read and write again. I said, maybe. And he taught himself. And that resilience made me think about, oh, we can all question uncertainty. And Gabriela Corral again thrived with that uncertainty of not being able to say for sure what the outcome of this experiment would be, aside from my mother's shock. And that uncertainty, being able to bring it into the classroom, bringing it to activities I do with students, and being able to embody my 5-year-old and say, don't be so sure. We can test this. Don't be so sure. My dad ended up reteaching himself how to read. And we weren't sure that this would cause harm, but we tried. And every experiment we do, I am really lucky that we have fantastic students here. And each one brings a slice of their curiosity, mists it, much like I misted my slices of bread, and we let it grow. And that's what Gabriela Corral did. And that ended up cross fertilizing maybe the other slices of bread, maybe our couch, but it had an impact. And it may not have been the traditional way of doing it, but it's sure an experiment that I enjoyed and to this day continue doing. So thank you. And I leave you with more.
Great. Thank you so much, Carlos. One more round of applause. Next, we have Matt Neubauer. Matt is a postdoctoral scholar who works on plant genetic engineering. He's been at NC State since April 2020 and has worked in the Alonso Stepanova lab in plant microbial biology, and now is in Jack Wang's Forest Biotech group. His current research focuses on implementing DNA-free CRISPR editing in woody plants to improve agriculture and sustainability. Matt is someone who's interested in the role genes play in determining plant and animal function and enjoys exploring how DNA sequencing technologies impact society. All right, let's hear it for Matt.
Hello. Thanks for giving me the chance to share my story today. For the last 11 years, I've studied plant genetics and I've given talks about plant genetics and plant mutants, but today I'm talking about my own genetics. And I kind of feel like today I'm the mutant that I'm talking about. I'm joking, but at the same time, kind of not really. So the story starts back in 2017 when I was a PhD student. I was studying Arabidopsis genetics, and I was, busy working in lab one day, browsing Amazon, and I saw that 23andMe kits were on sale for Prime Day. So I decided to just buy one of these kits that I had heard about because it was like 50% off, and I was like, how could you not buy this? So I bought the test purely out of curiosity. I didn't really expect any interesting information to come out of it. And when I got my results back a couple months later or whatever, it was kind of underwhelming. And I think a lot of people who take these tests, they get their test results back and they're like, eh. And that was how I felt. So I was like, well, it was only $100, who cares? But fast forward a couple months, for those of you who have taken one of these tests, you know that they will give you a list of DNA matches. And so as time goes on and more people take these tests, you get more and more matches added to your list. And so one day I'm walking home from lab and I get a message on my 23andMe app from someone who's a close relative. And this was weird because it was a name I had never heard of. Um, and so I read the message, and this stranger who has 25% of my DNA tells me that he had taken this test because he wanted to find some of his biological half siblings, because he had been told recently by his parents as an adult that he was conceived by sperm donation. And so he was either looking for his sperm donor or he was looking for one of his half siblings. So he was all excited to find me as a match, and I'm, walking home from lab and I'm reading this on my phone and I'm like, this doesn't make any sense. And so, at this point, I've been working studying genetics for years, and the data is right in front of me that, probably I'm donor conceived and I'm related to this person, but I was kind of in a state of denial for a little bit. So I messaged my half brother back telling him, hey, that's a crazy story you have, but I don't see how I fit into this because I was not donor conceived. Um, and so, as a couple days go by, I think reality kind of starts to dawn on me. I start to kind of look back on my past and my upbringing, and I can start to kind of pick out points where maybe my parents were sort of fibbing or telling little lies and things. I always looked a lot more like my mom and her family than I looked like my dad and stuff like that. Um, and so eventually I kind of get the nerve to talk to my parents about this, and they tell me that when they conceived me by sperm donation back in the 90s, they were told by the fertility clinic to keep it a secret. And that was really common advice back then, which I'll get to. I've personally experienced this as well. A lot of people have gone through similar experiences. Um, So this was not a very fun experience for me. I was, at the time I was probably 25 and I was learning this for the first time. And so as time passed, it took me months to kind of cope with this. I decided eventually that I wanted to take another DNA test to, like my half brother, see if I could find other half siblings. And it kind of became sort of a weird hobby of mine, I guess, because I've always been interested in genetics, and now I'm like studying my own genetics in a way. Um, so I started taking more tests and I started getting in contact with other half siblings. And it was kind of weird because there's so many different databases and whatnot. Some people had already contacted each other. And so you get added to a list and you find out you've got 10 half siblings on this website and 4 on this one and so on and so forth. And at one point I got in touch with someone and she sent me a link to a Google Doc or a spreadsheet where she was keeping track of everyone. And I was one of, at that point, maybe 12 people. And so we had columns of name, age, website that you were, uploaded to or whatever, and then like personality traits even that they were keeping track of. And so I guess for those of you who know me, I'm a very introverted person, and I'm looking at the list of my half siblings, and 12 out of 12 I identified as introverts. And so I was like, okay, this kind of makes sense. Um, as a geneticist, I wasn't the one who made that list. So I think this was something that even my non-geneticist half siblings, they were really interested in kind of exploring themselves this way. And so up to now, we have at least 21 half siblings. My Excel spreadsheet has a lot of people listed on it, but that's constantly being added to as more and more people take tests. So that's been strange. So about a year after my initial contact with my half brother, we actually found our donor parent. He had taken kind of an obscure DNA test that I hadn't even heard of, and he matched with one of my half sisters. And then she forwarded him all of our contact info, and he started to reach out to us. This was shocking to him. He was told back in the 90s that the most number of children that could be conceived from him was 12. And at that point, we'd already been many more than 12. So now we're like double that. So that was, I think, kind of shocking to him. But he's been really open to having contact with us. And that's been kind of a cool experience getting to know him. And so at this point, I guess up to now in the story, it's kind of a traumatic, sad story, but it's turned into something that's actually been more positive. Since learning about this, I've actually met with, I think, 4 or 5 of my half siblings, as well as met with my donor parent. And I've also had like internet communications with a lot of my other half siblings as well. And so for the first time in 2019, I flew out to New York to meet with my donor father, and I met with 2 of my half siblings, including the half brother who messaged me on 23andMe back in 2017 or 18. Um, and so it was kind of this weird experience of meeting a total stranger, but also someone, my biological father. I share half of my DNA with him, and we, in a weird way, did kind of instantly connect, even though we're strangers who grew up in different parts of the country and have very different experiences. Um, Physically, we look very similar, and we also had a lot of similar interests and things like that. And so that was kind of a fun experience for me, and I think for them as well. And so me and my donor father have become pretty close friends, and we keep in touch. We email back and forth. I've met with him 4 times now. Most recently, last fall, he was in Durham for his friend's wedding, and so we spent a day together hanging out in Raleigh. And then I found out that he has relatives in Chapel Hill. So I found out I have an uncle who lives like 20 minutes away in Chapel Hill that I didn't know about. And so like I said, this was initially really shocking, but it's ultimately, I think, had a pretty positive impact on me. It's sort of changed the way I view genetics, I think, to some extent. When you can actually, I'm joking when I say that I feel like I'm in a genetics experiment, but at the same time, it's hard to look at all of these people who are strangers to you and you find all the similarities, and it's like, maybe we did inherit that together. So that's been kind of fun. We've had a lot of, we've learned about a lot of commonalities that we share in terms of like hobbies and interests and personality traits. I was a cross country and track runner in high school, and a bunch of my half brothers also did cross country and track, as well as our paternal grandfather. So, in some ways, I think maybe that's something I inherited. Um, so, as I've kind of already alluded to, my parents were told not to tell me about the donor conception, and that was advice that a lot of fertility clinics were giving back in the 80s and 90s. And so, I think one of the takeaways I'd like to give from my story, I think, is The role that we as geneticists can play in sort of shaping public perception and also kind of informing those kinds of ethical, bioethical decisions. So I think back in the 90s, maybe no one would have predicted that DNA tests for $50 could tell you who your relatives were. They thought that these secrets could be kept forever. But obviously the pace of technological change really outpaced people's expectations. And I would like to think that if they had known where the future was headed, maybe their recommendations would have been different. Uh, and so, yeah, I just like to point out to all of you, I think we play an important role in kind of shaping the public's knowledge about these types of technologies and kind of informing people about where the future is headed in terms of biotechnology and genetics. And so with that, I'd like to say thanks for letting me share my story. I hope you guys found it interesting. If you have questions for me, I know this is like a very personal topic, but I really like talking about it, and I'm happy to take any questions you have. And if you have a crazy DNA test story, I'd also like to hear about it because this has become kind of a weird hobby of mine at this point. So yeah, thanks.
All right, one more round of applause for Matt. That's a great story. Our third storyteller is Isabella Livingston. She is a fourth-year PhD candidate in the genetics program and also the recipient of the GAN Molecular Biotechnology Fellowship. Woohoo! Isabella grew up in Portland, Oregon, and studies how various genetic and genomic tools can be applied in wildlife health and conservation. Her focal research groups are the Galapagos sea lion, the California sea lion, and the American black bear. She's passionate about wildlife and conservation and is eager to work with as many species as possible. When not in the lab, Isabella can be found in her mini zoo with her Labradoodle, her bearded dragon, and her Maine Coon. All right, let's hear it for Isabella.
Thank you for the introduction. So they say that the average person hears no around 1,000 times a year. And I would argue for people in STEM that that number is probably higher. And no is often given a negative connotation, and rightfully so. But throughout my journey, I've learned to harness the power of no as a motivator, and I've learned to pivot when faced with rejection. So one moment can set the course for your entire life. And for me, that moment happened when I was 5 years old. It was a normal evening in my household, and my dad, who was in charge of my two older brothers, and I decided it would be a great idea for us to all watch his favorite movie, Jaws. Now, he watched me that whole time, kind of waiting for that inevitable terror to set in, but he was shocked when it never came. Actually, I was enthralled by the shark that was terrorizing Amity Island, which I'm aware that makes me sound like a very strange and creepy 5-year-old. But I wasn't really interested in the plot. I was more interested in the animal itself. See, my parents had told me my love for animals started when I was very, very young. My cribs and my beds were always buried in stuffed animals, and I always wanted to sleep with the dogs and the cats in their beds. But I didn't really have a lot of exposure to sharks. This was the first time I was seeing one. And I remember my dad telling me about Peter Benchley, the original author of Jaws, and how he felt so bad after he saw what his creation did to sharks' reputation that he dedicated the rest of his life to studying them. And I couldn't understand why people hated sharks so much. I mean, I'd seen The Lion King, I was well versed in the circle of life, and I couldn't fault that shark for needing to eat. But really, I credit this as the spark that set me on the path that I'm still following to this day. I wanted to be a shark researcher, and I wanted to show the world the love and the respect that I felt that they deserved. So from this point on, sharks kind of became my identity. I was known as the shark girl, and still to this day, when people don't know what to get me as a gift, they turn to anything shark related. Every homework assignment, every project, every art piece, if I could find a way to incorporate sharks, I did, even when my teachers really didn't want me to. And I spent all my time trying to learn about them. But the first time that I realized that studying sharks wasn't going to be as straightforward as I thought, and the first no that I received pertaining to this dream was when I was 12 years old. I was interning with a marine biologist who was working on a project studying whales, and she told me that there weren't a lot of jobs that were solely focused on sharks, and that I probably wasn't going to be able to do what I wanted to do. And of course, to a 12-year-old, this is devastating, but I thought about something my parents had always told me, that if you wanted something badly enough, you couldn't just expect it to happen. You had to make it happen for yourself. So, like Ross in that episode of Friends when he's moving the couch up the stairs, I needed to pivot. So I broadened my goals from being a shark researcher to a marine biologist. And then I spent my time studying marine animals and biology and all I needed to do academically to become a marine biologist. And I wouldn't say I was necessarily gifted in math or science. It was something I really had to work for because I knew that shark research and animal research was going to involve STEM. And around the time that I was looking for colleges, I was hearing more about issues like biodiversity loss and endangered species, and it really struck me that my peers didn't seem to be as bothered about these things as I was. And I revisited that idea of being bothered enough or wanting something enough that you needed to do it for yourself. And so I started to look for programs in zoology, thinking that maybe I shouldn't restrict myself to certain species when so many of them needed help. And I started school at San Diego State University as a biology major with an emphasis in zoology. And I vividly remember during orientation for the biology program, before classes had even started, the professor who was leading it was telling all of these students how hard it was going to be, how most of us were going to fail classes, and most of us were going to change majors. But by then, I'd heard so many times similar things, and that it was okay for me to give up on my dreams. But all of this served as motivation and fueled my determination to prove people wrong. And so I had to put a lot of work in. I had to sacrifice late nights doing fun things for being in the library and trade in my afternoons for office hours, all while working 3 jobs to try and support myself. And I remember during one of these office hours, a professor I really admired told me that people didn't really care about animal research. I was going to need to go to grad school. It was going to be hard, but I needed to let go of my animal dreams and focus on humans if I wanted to be a successful scientist. But I couldn't fathom working as hard as I had to get where I was, only to give up the thing that really inspired me. Of course, at this point, I loved science. I'd grown really interested in it, but the only reason I was in science in the first place was for wildlife. And so I thought maybe I might need to pivot again. And I looked for research experience in any lab that had an open position for undergraduates. But one thing led to another, and I ended up in a marine microbial ecology lab. And as if by fate, I ended up on the only non-microbial based project in that lab, and it was one involving sharks. Now, this was my real first exposure to research, and I felt more motivated than ever to start looking for grad school. So I sent out hundreds of cold emails to potential advisors, most of which were met with silence, or as many could probably guess, no's, and there's no funding for wildlife. But eventually I did find a professor at UMass Amherst who was doing shark adjacent research. And we spoke for months, coming up with a project that I could take on, and I thought I was set. But in December of 2019, when it was way too late for me to find another advisor for the next academic year, he told me he couldn't take on new students anymore. And it felt like the ground had fallen out from beneath my feet. I had no idea what to do. I had never thought about taking a gap year, but again, I needed to pivot. So I graduated in May of 2020, and I found a position as a research technician in the lab of Dr. Forrest Rower on his project studying and swabbing the urban environment and looking for the presence of COVID-19. And I was back in the world of genetics and genomics, and I realized the power of these tools and how they could be applied to any organism. And so I started to look at grad programs again, looking for wildlife genetics. But I was warned that I shouldn't be looking for a specific study subject. I should instead focus on a tool or methodology that could maybe one day again be applied to animals. But again, I'd never really thought about a specific method or tool. I had been more focused on the study subject itself. And I didn't know what to do or what to choose, and no one I talked to in wildlife seemed to have anything reassuring to tell me. But I set out anyway. And fortunately, I found the GGS program. And let me just say how amazed I was about hearing all these animal-related projects that never came up on any Google search I did. But through rotations, I found my PI who shares my enthusiasm and passion for wildlife, and he gave me the space and the support to not only take on a project, but create a project that inspired me to work every day. So now I get to work with species like the Galapagos sea lion and the California sea lion and the black bear, and show how well genetic and genomic tools can work in questions of wildlife health and conservation. And I get to go to places like the Galapagos and get up close and personal with bears and sea lions and iguanas and wolves and more wildlife than I ever envisioned I'd be able to work with. And each time that I do this, I think back to the way that I felt as that 5-year-old girl watching Jaws and just dreaming of a life surrounded by animals. And it makes all of the rejections and obstacles that I face seem so trivial. And I know I'm not working with sharks, but I'm surrounded by animals every day, and I'm in a position to address those issues that bothered me, like conservation and endangered species. And I'm fairly certain sharks are going to find their way back into my life. So I know this story is repetitive, but I wanted to at least show a little bit how often I was told to give up on my dreams and how often I had to be my own advocate to make sure those dreams became a reality. And this is by no means an exhaustive list of all the times I was told no. But each time I was told no became an opportunity for me to pivot and to find another way forward. And I've also had opportunities to work with countless aspiring STEM students and hear their stories of the rejections and the obstacles they've had to face. And if anything, I hope that by sharing my story, I can show people who might be in the same boat that, as cliche as it is, truly anything is possible if you work for it. If you're passionate about something, there are ways that you can make it happen. It won't be easy. You have to make sacrifices. You have to work hard and be resourceful and face many, many obstacles, but it is always so worth it. Thank you.
All right, let's hear it for Isabella one more time. All right, so we're just going to take a slight pause before we hear our last two speakers. And I just want to highlight that we do have a workshop series coming this fall on how to tell personal stories. You don't have to have a story all dialed in. You can just have a moment where something changed in your life. Or maybe something you wanted, but there was something that stood in the way. Just bring those just nascent ideas, and we are going to help you shape out a story so you can also join all the storytellers up on the stage and tell your stories. Our plan is to start having regular storytelling evenings, and so this is going to be sort of the lead up to that. So look at more opportunities for that. So we have two more. Are you all ready for the last two? All right. So our next one is Emma Droste, and she's a third-year PhD student in genetics and genomics program, and she also received the GAN Molecular Biotechnology Fellowship. So woohoo! She received a bachelor's in genetics and master's in STEM education at NC State. She studies the connection between genetics and neuroscience using fruit fly brains. She also uses her experience in education to help adapt scientific research into lesson plans accessible to K through 12 students, classrooms. In Emma's free time, she has lots of hobbies that include reading, fiber arts, theater, and I do want to say she's also thinking about writing a book. So without further ado, Emma.
I'm sorry. I'll just leave this up here the whole time. Yeah, I'll get to the book after the thesis. So when I am passionate about something, my eyes light up. And this is something that people have told me all my life, that when I talk about something that I really love, like my work or the educational outreach that I do, or a book that I really love, my eyes light up. And my passions in genetics and education that I'm currently pursuing didn't really come out of nowhere. This is also an origin story. It probably started all the way back in high school biology. We're learning Punnett squares and pedigrees, and fractions are making sense for the first time in my entire life. And at the same time that I'm learning genetics in high school, we are also learning something new and potentially scary about my little brother. He's in the middle here. And that is that he has a diagnosis of neurofibromatosis type 1, which I'm just going to call NF1 because neurofibromatosis is a big word. So for those of you not familiar with NF1, it is a genetic disorder characterized by mutations in the NF1 protein that has a wide range of effects, including tumors wherever there are nerves in the body, potential tumors wherever there are nerves in the body, reading, writing, spelling difficulties, scoliosis, Lish nodules, and cafe au lait spots, which are both pigment things, which was actually how we found out my brother had this. He's mosaic, which is really cool, and he has all these like markers on the right side of his body. Very like none of the Lish nodules on his left, none of the cafe au lait spots on his left. It's mostly on the right. And yeah, because it's this disorder with a wide range of effects, people in the scientific community really focus on that tumor aspect, which makes sense. It can be really scary to potentially have these tumors because they end up in places like the spinal cord or the central nervous system where it's very hard to operate. And they are these complex tumors where the neurons twist on themselves. And so you hear about that and you're like, that's scary. And my mom, I remember, was freaking out about it. And she was like, oh my gosh, you and your sister have these cafe au lait spots. We should get you tested. And while my family was very scared, I was very curious about this thing. And because I couldn't really see it hurting my brother at that point. But I remember telling my mom, like, Mom, it's autosomal dominant. It's okay. Like, if me and my sister had it, we would have it. But we don't. And my brother's this really cool mutation who only has it on the right side of his body. So we're good. And so my brother gets screened every 6 months, and this tumor aspect, which is where a lot of the research dollars goes to, is actually not the part that touches my family the most. The experience that my brother had most of the part and the struggles he mostly faces are actually with that reading, writing, spelling difficulties. And to hear my brother explain it when we were kids, when he was learning how to read, he would spend so much time sounding out the words, understanding the letters in the words, and sounding out each word in a sentence, that by the time he got to the end of it, he couldn't tell you what the rest of it said. And that was fascinating to me. Like how can what may just be a point mutation in his NF protein have such a wide effect and have such an effect on the way his brain is wired that he has this difficulty. And to be clear, my brother is smart as a whip. He doesn't like to say that he is, and I don't like that when he calls himself dumb because he can't read. But he's a senior in college right now. He's so smart. You tell him something verbally and he can quote it back to you in like a month. But ask him to read it, and that's really difficult. And so the following summer, after we get this diagnosis, and all of a sudden, like a lot of his difficulties with reading are making sense now, it was the first summer he was going to have summer reading. And out of the selection of books, none of them had an audiobook, or the one that he really wanted to read didn't have an audiobook. And so me, being the avid reader and performer that I am, I was like, I'll do it. I'll read the book. I'll record it for you, and you can listen to it back when you have time. And that, then you can do your reading and just get through the summer reading assignment. And so I'm reading this book. I can only read it in 2, 3, maybe 4-hour chunks because I only have so much time. And my voice actually would hurt because the story is about this girl stranded on a raft, and she's so dehydrated that she's hallucinating that the captain of the plane that crashed is in there coaching her through this survival experience. And something happened for the first time that I noticed, which is that my brother was really getting into the book. And he was like, Emma, read the next section. Get to the next section. Please record it. I want to know what happens next. And I saw a little bit of that spark in his eye, that he was really enjoying this experience. And I was just like, wow. In this moment, like, it was almost like my passion was contagious a little bit, something that I really loved was something that I could get him to love just by making it a little more accessible to him, making it putting it in a format that's accessible to him. And that has kind of been one of the formative experiences that has driven me through my work here. I drove me through my bachelor's in genetics here. It is what sparked my want and drove my energy to actually get my master's in STEM education. And now it's what's driving me to study this intersection of genetics and neural activity, which if you want to stop by my poster during the next session, you can. I'm all the way in the corner. But yeah, that's my experience with just with making education accessible. And I truly hope that I can do that for the rest of my life and make my passions contagious. Thank you.
All right, let's give another big round of applause to Emma. A powerful story. Last but not least, Bruce Schulte. Bruce is the new department head of biological sciences here at NC State. Prior to coming to NC State, he was the associate vice president of strategy, performance, and accountability at Western Kentucky University, where he also served as the department head of biology. His research is in areas of animal behavior, chemical ecology, and conservation. I see that theme. Bruce has studied elephants for the last 30 years with an increasing focus on mitigating human-elephant conflicts using a conservation behavior approach. And I will advance the slide. I'm so sorry I didn't do that last time. Do what you can, okay, go to the next one. I'm going to the next one now. There we go. All right, thank you. Let's give him up.
Great. Thank you, everybody. Pleasure to be here today. Thanks, Jeff, for inviting me. I am extremely happy to be the Department Head of Biological Sciences. I've been here about 23 days on the job, and every day that I get the opportunity to speak to another one of the faculty of biosciences, I just find out how tremendously wonderful people they are, and brilliant scientists, excellent educators, and I couldn't be in a better place. So thank you for making this part of my career. I'm very appreciative. On this story today, and also I should thank Jeff directly for the GGA invite and for feeding me today, even though I was late on my order of vegetarian lunch. We also had the coup of getting Jeff in biosciences now, so I thought that was a pretty good deal that I didn't ask for in my startup, so good job. I've been studying elephants for a while, and if you look at the slide up here, you'll see cartoons in the upper corners by Gary Larson, who is the wildlife biologist's favorite cartoonist and friend. You can always find a Gary Larson cartoon that will suit what you're doing. And Isabella, hopefully you found some yourself. The logo there is the no-go logo. You're not allowed to have a logo as a department, but the department does have their logo there. So that's what's up there as well. And then the picture is some of my friends. So when we think about elephants, most of us in Western society anyways, think about elephants as these sort of romantic creatures that we've heard about for a long time. We very often have grown up with them through various stories and movies and things like that. They're thought of as kind of curious creatures, intelligent creatures, kind of fun creatures, that sort of stuff. We have a very positive attitude towards elephants, which is great because clearly they're extremely cute. And it's hard to turn away from a baby elephant, even though they weigh about 250 pounds and can knock you over without any problem whatsoever, even if they don't intend to. But they're adorable animals, no doubt about it. When I was young, I grew up in upstate New York in a small town riding horses, and I don't think my parents ever thought that that would turn the skill to be able to ride an elephant, but that did happen to me, and it's happened numerous times. I also have had horses my entire life, and that's one of my daughters and I riding horses in Kentucky, and then the other daughter riding an elephant in Zambia. So I think it's genetic, which is part of this conference, right? So maybe there's a gene-environment interaction going on. But that's not where I started. When I was in upstate New York, in landlocked little town in upstate New York, Jacques Cousteau was my hero, and I wanted to be a marine biologist. And I knew that from about 7th grade on, and really never wavered. Went to college at William & Mary and took all the marine courses that they had, and went out to graduate school at the University of Southern California. What I found out, though, is when I was at the University of Southern California, I was working, diving all over the world and supposed to do research out in the islands off of India, that they thought I worked for the CIA and would not give me a permanent visa to live out on the islands out there. I can't tell you if I did work for the CIA. I'd have to kill you if I did. But what happened was I wound up getting my master's and then turning back to working with my brother in a resort and leading trails, creating a nature hike to go to beaver trails and beaver mounds and beaver sites and all this stuff. And eventually finding a professor in the environmental school in Syracuse, New York, who had an NSF grant to study the chemical communication of beavers. And that's what I wound up doing. Still, perhaps my favorite animal. But it doesn't get you to elephants. Now, the interesting thing is that beavers in the Pleistocene were about the size of today's black bear. So there were some big creatures in my past and then wound up being in my future. So they really are big. The video's gonna work. Maybe not. Too bad. All right. It didn't blink. So this guy is called Valley M. He's one of my favorite elephants. He's from a park that we did work in for over a decade in South Africa. And what the video would show is you walking up him up next to our vehicle. So he was literally about here, and he filled the entire windows of the cars. And I had 3 students in with me, and he was in must at the time, so he smelled pretty rank. And I could have reached out and touched him. Oh, is it moving? It did? It did. Maybe. Oh, here we go. All right. Yeah, so this guy's big. Yeah. And the window's down because I wanted them to get the full smell. And so there he is. And he did stop and he checked out our car and he knew me. So we had a good relationship again, as long as I didn't touch him. Because if I touched him once, that would have been the end of the relationship, because it would have been the end of me. But where I really got introduced to elephants on a personal basis is when I did a postdoctoral work, and I had 2 interviews. One was with John Vandenberg. Anybody ever heard of him? John was a professor here, and I came down here on a bus from Syracuse, New York, and interviewed with John, and John was studying small rodents, and that could have been my postdoc. And then the other postdoc that my professor told me about was this woman that was studying elephants out in Oregon at the Beaverton Oregon Institute in Beaverton. She'd never had a postdoc. And so I flew out there with my wife, and we convinced her that I needed to study elephants because of their relationship with beavers. And fortunately, she really reveled my professor, who did his PhD under Konrad Lorenz. If you don't know who Konrad is one of the 3 Nobel laureates to start the field of behavior called ethology. So I was fortunate enough to be the academic grandson of a Nobel laureate in the field of behavior. So anyways, we convinced her to take me on, and we were studying Packy is an elephant that was born in the Oregon Zoo. It was unusual because both his parents were captive born. He was probably the first elephant born in captivity from 2 captive born parents. But she was studying chemical signals in elephants and found out that one of the pheromones in elephants that females release to show that they're coming into estrus is the same chemical of over 120 species of lepidopterans, butterflies and moths. We think it's probably convergent evolution, though. We don't think elephants evolved from butterflies and moths. So. But I had to bring a little more genes into this story, right? So, of course, elephants use chemical signals because proboscis, proboscidea, the nose, they have this famous nose. Amongst many famous noses, they are perhaps the most famous of all. And this is really why all those kid stories are so cool, because think about what Dumbo does that he thinks he can do to fly, right? He's got to hold that feather. Yeah, so tremendous noses. And so we studied chemical communication for a long time. And then to bring the genetics into the story, I'm not supposed to show data, so that's not my data. But other people have now shown that they have the highest number of olfactory receptors genes of any mammal that's been studied. They are, in fact, probably the best smellers on the planet that we know of so far. They kind of put mice and hound dogs to shame. So they're really good at this. And we try to use this information because I'll tell you, for years and years, trying to get NSF grants. And remember those pages on NSF where you have to check off like what your animal is, and then they give you all these model species like C. elegans and mice and stuff like that. And I always had to click other and then add elephants. And then you'd go to the NSF panel and they'd kind of laugh like, elephants, the model species, sure. And so you go, yeah, come on, they are. They're the best ones for studying this stuff. Still trying to talk NSF into that. But during that time, I didn't give up on marine biology because I actually got back into studying manatees, which, if you don't know it, are the closest living relatives to elephants, those in hyraxes. And so I studied them for a long time when I was down in Georgia, and kind of gave that up when I moved to Kentucky because of the distance and the logistics involved. But it was great because it got me back into that world of studying a marine organism again. But then my research sort of turned because of the personal stories that I was hearing. So we worked in sub-Saharan Africa and a number of countries in southern and eastern Africa. And those people don't love elephants. They didn't really think much of Babar or, Dumbo or any of those, because every day they live with them, they're under the threat of being killed by them, of having their crops eaten by them. And the elephant does get into their kitchen, and the elephant does cause trauma for them. So they're not very happy to have elephants around. It's a very, very different story. And the story that as Westerners that we were alerted to for pretty much the last 50 years is the poaching story, in which Western societies and Eastern societies have valued basically a tooth, which is kind of silly, and killed an entire animal to saw off a tooth and use it for all sorts of obscure things and medicines that don't really work and all that kind of stuff. This is not an angle that I went into, although it's definitely an angle in all the places I've worked that I've had to be very aware of, because it's extremely dangerous in certain places to be working on elephants in the field, knowing there are poachers around. So you become very good friends with rangers that become your best buddies, and you bring them with you everywhere you go. And they have the guns, not for the elephants, but for the poachers. But they actually have used elephants to try to find poachers and to actually sniff out poachers. So that's been sort of an interesting application of the chemical signaling part of elephants and their enemy, if you will, the poacher. But when I've been in Kentucky for the last 15 years, the old story of the Hatfields and McCoys, which if you know that story on the eastern side of Kentucky, along the Tug River in West Virginia, the Hatfield McCoys in the 1800s were two families that fought each other for lots of ages and ages for no good reason, and never really figured out how to get along. And that's kind of how it is with humans and elephants in both Asia and Africa in many societies. And so we sort of try to think about the Hatfield-McCoy feud and said, how do we resolve something like this? What do we have to do to try to get them to get along? And it's a very real feud, because what you see up here in the picture is a field of maize, or what we call corn, and other plants. And then on the other side is what happens after elephants visit that field in the nighttime. They basically can wipe out a farmer's entire food and income in an evening. So the feud is real, and we have to figure out a way to sort of resolve it. And again, we understood this by going and working with the farmers, visiting their villages, staying with them at times, and really getting to know their story and understanding what their needs were. And most of this damage is done at night. And so farmers will literally go out at night trying to scare elephants off. Now, being in the bush at night is not a good idea anyways, but when you're doing it with knowing elephants are around and the most you have is fire or sticks or that kind of stuff, not a great idea. But what it also leads to is in the light of day, they go out and look for elephants to injure them or sometimes kill them in retaliation. Not necessarily even the same elephant that came into their field because they usually don't know. So it becomes a huge conservation issue because not only do they not want the elephants there, they actively work to hurt them. And so that story came to us of like, okay, we've got to figure out a way for people to understand elephants so that they can live with them. And so we've been involved on all sorts of projects. That's me helping to build a fence that we're using local sources of materials that are affordable and practical for the farmers. We know we can put big electrical fences around places and keep them out, but the farmers can't afford that. It won't keep up. Solar batteries will be used for something else. So we have to use things that they can actually afford, that they will actually upkeep. And then the plant part that you see there, the maize growing, those are known as zypits. So they're climate smart agricultural approaches to trying to increase the productivity of corn. Hopefully it grows faster. These are very drought-ridden areas. So we were trying to adapt things that they already knew and then use experiments to figure out what's the combination of sort of protecting the fields and growing the crops that the farmers can afford. And it's a little bit like the story of the three little pigs. You know, if the wolf doesn't come very often, you can probably make it okay in a straw hut. But if the wolf's coming around, you kind of wish you had that brick house. So we have to figure that out with, or almost on a village by village basis. And the only way you can get them to invest in protection is they're really afraid the elephants are going to come. The other thing that they do is go into their habitat and take wild food from the wild, which actually exacerbates the problem. So we get involved in the classroom trying to get the children to understand how to grow up in an appropriate way of not only not getting hurt by elephants, but living with elephants in a way that they don't harm the wild areas around them so that the animals have some place to live. So my story goes back a long time. Those are our two daughters when they were young. My wife is actually a potter and does a lot of work with endangered species in her pottery, including elephants. Lots of students involved. We've now turned to going back to studying Asian elephants again, like I did with Paki, and have a student in Sri Lanka who just came back at the bottom there. And I've been fortunate that I haven't totally given up my marine origins because all the animals I study like to take a good swim now and again. So thanks again for your time. Enjoyed being here. Appreciate it.
All right, that concludes our storytelling event. Let's give a huge round of applause for our 5 amazing storytellers. Thank you so much. So we are actually going to take a break now, but what I recommend, highly recommend, is that you spend some time checking out these amazing posters. Voting will be on either side, on stage left and stage right. And we are voting on one vote, but for 3 posters that you think are the 3 best posters. And that is going to be done at 3:15. We will Rebecca and I will go in the back and we are going to figure out who the winners are and we will announce them right away. This is like instant gratification. So definitely spend some time, get your coffee, and talk to these amazing poster presenters. Thank you.